Hello lovelies, in this video we're going to be going through your AQA A-Level Biology paper 3. This is the big one. So this is a synoptic paper with the essays at the end. So we've gone through both essays at the end, not only the example essay, but how to plan and prepare for it, so how to write a mini plan in the exam, how you can link all the things together so you can take this skill and use it in your real exam.
The kidney is responsible for excretion and control of the water potential of blood. In the following passage, the numbered spaces can be filled with biological terms. Write the correct biological term beside each number below that matched the space in the passage. So this is basically a gap fill, but with no words. So this is an A-level gap fill. And that means there might be some alternative options to the words, but most of the time we're going to have to make sure we get the word right and we spell it right. So blood enters the glomerulus from the something artery through the afferent arteriole. So it's something that's entering the kidney, entering at the glomerulus point. That is a main artery, and obviously it comes through the afferent arteriole. So it comes from a certain artery and then branches into the afferent arteriole. So we can't put that as an answer. But what is the main artery that brings blood to the kidney? It is the renal artery. Then it says blood flows through the knot of capillaries and leaves through the efferent arteriole, which has a narrower what than the afferent arteriole. So this is where it makes higher pressure because it has the narrower lumen, which is the hole, the space in the middle of the blood vessel. This creates a high something pressure, as I just said, which forces the small molecules, including glucose and water and ions, through the gaps. So what type of pressure is the type that forces these small molecules out? Just like in tissue fluid, it's hydrostatic pressure. So we're forcing ions through the walls of capillary endothelium into the lumen of the something capsule. So this is where the liquid is going when it's being forced out of the blood vessels in the glomerulus. It's forming filtrate, and then it's going into the lumen of the next part of the structure of the nephron, which is the something capsule. Hopefully we know that's the Bowman's capsule. That is the name for that piece of the structure. Should have a capital B because it's named after someone. This is the process called something. So what does this whole process describe? This removal of substances from the blood and then collecting them as filtrate into the Bowman's capsule, where all of these water, mineral ions, glucose and everything, amino acids is being forced out. It's the first stage of the process in the kidney. And think about this idea that obviously we're like filtering all of this stuff out of the blood, but we're kind of forcing out like through a filter. It is known as ultrafiltration. Okay, so we have to get these words in this right order. You could have had a slight alternative for lumen, which is about diameter, because think about the diameter of the lumen. And everything else needs to be, you need to have the capital letter and the apostrophe for Bowman's. You need to have ultrafiltration, not just filtration. And yeah, it's allowing phonetic spelling. So as long as it doesn't look like another science word that could be a wrong answer, then it doesn't necessarily matter about the spelling, but it does matter for the capital letter and apostrophe for mark number 4. The graph below shows the concentration of the filtrate and the concentration of glucose in the filtrate in the different sections of the nephron after the glomerulus. So we've got the PCT, we've got the loop of Henle, and then we've got the DCT and the collecting duct. Explain why the change in glucose does not affect the concentration of the filtrate in the PCT. So let's think about what happens in the PCT. It's where the water, the glucose, the ions, the amino acids, everything is all absorbed back into the blood that is useful and important that we can absorb. But it's asking, why does us decreasing that concentration of glucose in the filtrate, so why does reabsorbing that glucose not affect the concentration of the filtrate? So if you're losing glucose, why doesn't it get more dilute, for example? Because obviously we're also losing ions and amino acids and everything else, but the main reason why is that water is also moving by osmosis. So effectively at this point, the concentration of the filtrate in the PCT stays in equilibrium with the concentration of the blood. Because at this point, we're just reabsorbing most of it by diffusion and then some of it by active transport, just to try and remove all of that substances. So water will move by osmosis with all of those materials as well. So although you're losing all of those substances that would normally kind of make the concentration more dilute, you're also, the water that would be left behind to make the concentration of the filtrate more dilute is moving with them. So we're not getting loads of water left behind and therefore it being dilute. Explain how the graph shows that this person must have been secreting ADH. So ADH is a hormone which increases the permeability of the collecting duct to water. This means that more water is reabsorbed into the blood by osmosis. So that would mean there'll be a more concentrated filtrate. And we can see from the graph that the concentration does increase. The concentration in the collecting duct increases as ADH makes the walls more permeable to water, so more water leaves by osmosis. So I've got kind of my evidence from the graph, which is that the concentration in the collecting duct increases, and then my explanation of that with ADH included. Some people who have diabetes do not secrete insulin. Glucose can be present in the urine of people with diabetes. Explain why one symptom of diabetes is producing higher volumes of urine. So, higher volumes of urine would mean there's more water in the urine, okay, because it's more dilute. This must mean that less water is being reabsorbed by osmosis from the collecting duct, which is the question we were just basically writing. So why does that happen? Why is glucose in the blood, or glucose in this case being present in the urine of people with diabetes, a clue to explain why less water is being reabsorbed by osmosis? So there's glucose in the urine, which means there's glucose in the filtrate because not all of it would have been reabsorbed at the proximal convoluted tubule, which is where it should have been. If you've got too much glucose in the blood, that won't happen. So if glucose is in the filtrate, it's going to lower the water potential of the filtrate. And that means there's going to be less of a water potential gradient between the blood and the filtrate, which is what we need in order for water to leave the collecting duct by osmosis. So this makes sense. If there's less of a water potential difference, less of a difference in the gradient, then less water is going to move by osmosis back out of the collecting duct and into the blood. Glucose is present in the filtrate, which lowers the water potential. There is less difference in water potential between the filtrate and the blood, so less water leaves the collecting duct by osmosis. Gas exchange in the lesser pondweed, Kitten bacillus, was measured by placing shoot sections in phenol red indicator solution. In the experiment, researchers put the same amount of red phenol solution into 4 tubes. They sealed each tube with a cap and let them sit for 10 minutes. Then they put plant shoots into tubes A to C and gave them different treatments according to the table below. After that, they placed the tubes in front of a lamp and left them there for an hour, as shown in the diagram. So the 4 different treatments, we've got the plant with just in the tube, uncovered. We've got the plant in the tube, but this time covered with tin foil. So this is obviously trying to replicate darkness, no light. Then we've got the tube covered with a fine mesh. So it's translucent. So that means that some light will get through, but it's not going to be as the same as the treatment in A. And then we've got no pondweed and obviously no covering on the tube. So this is our control tube, effectively, the only one that doesn't have the pondweed in it. Explain the purpose of tube D. So, as we've just said, we're just talking about the fact that that's going to be our control. But you can't just say it's the control. That would be kind of stating what the tube is. It wants us to explain. So why is that our control and what's it for? So because it doesn't have the pondweed in it, and obviously hopefully we can tell from the table that we're looking for a colour change with this indicator solution, it's going to be to show that any colour change in the solution is because the plant is there. So having the plant not there shows you that the indicator is not just going to change on its own. And that's the kind of the bit that's important. We need to make sure that that colour change is due to the thing we've added, which is the pondweed, and not anything else, not that it just would change if you left it on the side. So to act as a control to show any colour changes are caused by the pondweed. Explain why students use the ruler. So again, we've got a piece of equipment. It's asking us to explain it. So why are we using it? What's the point? Why is it important? So obviously it's to keep the same distance between the lamp and all of the tubes. So they must have measured that they all had the same distance from the lamp. But why is that important? So obviously the light intensity and the temperature is going to be the same from the lamp in theory if you're the same distance away. And now the reason we're talking light intensity and temperature is because if you've noticed, this question is about gas exchange. It's not about measuring the rate of photosynthesis and it's not about measuring the rate of respiration. It's about this idea of gasings being exchanged. So we're looking at, in a plant, therefore, both respiration and photosynthesis will contribute to the exchange of gases. So this isn't just about measuring the rate of photosynthesis. So it's not that we only care about light. We're going to have to focus on temperature as well, because both of these can affect the reaction rate of both photosynthesis and respiration. So temperature and light affect photosynthesis, and temperature will affect respiration. So that the temperature and light intensity are the same for all tubes, as these can affect the rate of photosynthesis. And you can see respiration here as well. As long as you've got temperature and light intensity in there, then even if you just didn't mention respiration in the second part, then that's fine. But you have to mention temperature at some point in your answer to get both of the marks. The colour changes of phenol red are outlined in the table below. So we've clearly got yellow is showing a more acidic pH, and red is showing a more alkali pH, and orange is around neutral in the middle. So it wants us to explain the results in tubes A to C in terms of the colour changes of phenol red. And so we need to give reasons for the colour changes we can see. Clearly orange is the start point, and obviously in the one that didn't have any pondweed, it stayed orange. So it stayed roughly neutral. So we've kind of got an indication that that is obviously what happens if there is no change, if there's no gas exchange, nothing happening. And so if we look at our other answers then, so A is red, so it's going to be more alkaline. B is yellow, so it became more acidic. And then C is orange, so it stayed relatively neutral. There wasn't really any change. So if we think about the processes that are going to be happening in each of these tubes, in A, there's definitely going to be photosynthesis happening because it's uncovered, so there's going to be light. In B, there's no light, so there's no photosynthesis happening, so therefore it's just respiration only. So in A, obviously we've got photosynthesis and respiration, And then in C, again, there's light, and so we can have respiration and photosynthesis, but it's a lower light intensity, or there'll be less light than in A, so therefore less photosynthesis than in A. So how can we link these processes to whether something's more acidic or alkaline? Obviously, carbon dioxide dissolved in liquid makes it more acidic because it makes carbonic acid. Carbon dioxide being used up in the water, any dissolved carbon dioxide being used up in the water, will mean that then we're removing acidic ions, so therefore it's going to make it more alkaline. Carbon dioxide is used up in photosynthesis because it's absorbed by the plant, and carbon dioxide will be being produced in respiration. So when we see a yellow colour, we see it more acidic, there must be more carbon dioxide in the solution, so therefore less photosynthesis is happening. And when we see it's red, there's less carbon dioxide dissolved in the solution, so more photosynthesis must be happening to absorb it and take it out of the solution. So if you haven't guessed already, this question is about the relationship between carbon dioxide, respiration, and photosynthesis, and the rates and how they change. So when there's more carbon dioxide, less photosynthesis is happening compared to respiration, or the rate of photosynthesis is lower than the rate of respiration. When there's less carbon dioxide, more photosynthesis must be happening than respiration, or the rate of photosynthesis is greater than the rate of respiration. And this is the patterns that we're seeing, and this is the explanation for these colour changes. So in tube A, there is the highest light intensity. So the rate of photosynthesis is greater than the rate of respiration. So carbon dioxide in the solution is absorbed, reducing the acidity. Now there is just a mark in the mark scheme you can see for explaining this whole idea about carbon dioxide dissolving produces an acidic solution. So I've mentioned that at the end of this point, so that counts as that mark. In tube B, there is no light, so the rate of respiration is greater than the rate of photosynthesis. You could even say no photosynthesis is happening in tube B. So more carbon dioxide is dissolved in the water, increasing the acidity. In tube C, the rate of photosynthesis and respiration must be equal because there's obviously no change in the colour, so therefore there's no increase or decrease in carbon dioxide. Its net is overall 0. So the pH remains almost neutral. So this is very much putting all your knowledge together from the understanding of photosynthesis and respiration, the understanding of how carbon dioxide can affect a solution if it's dissolved in it, and sort of matching up this idea of the table and the colours and the indicator level to kind of figure out what the pH is and going on. It's an analysed data question, but it's also got to allow a lot of that knowledge of the relationship between carbon dioxide, photosynthesis and respiration. Explain why, when the pondweed respires, the oxygen concentration in the water around it decreases. So it's an explain question, so we need to give reasons why. So we're giving reasons why, we're explaining why oxygen is used up in respiration. So obviously this is going to require us to talk about how and where oxygen is actually used up in the reaction. What role does it play in the reaction? And hopefully we should know that obviously the part that oxygen plays, the reason why we need oxygen in respiration reaction is because it acts as the final electron acceptor. This is a nice kind of bit to kind of learn, because any explanation or any question that's asking you to explain how oxygen is used in respiration, or why oxygen is used up in respiration, or what happens and why does respiration stop if there's not enough oxygen, this is always the same answer, because you're basically just describing how oxygen is used. In aerobic respiration, oxygen is needed to act as the final electron acceptor of the electron transport chain. It combines with H+ ions and electrons to form water. Students set up an experiment to investigate the effect of the direction of light on the growth of the stem and roots of cress seedlings over 48 hours. They set up 2 treatment groups, each with 60 seedlings. Group A was grown for 24 hours in the dark, and then 24 hours with a single lamp placed to the left of the growth chamber. Group B was grown for 48 hours in the dark. So we've got no light entirely for Group B, and we've got no light at the start for Group A for half the time, and then the other half of the time they were exposed to a light that was coming from one direction only. The students measured the growth of stems and roots in each seedling in both groups. The results are shown in the table below. State the type of hormone that regulates these plant responses. So clearly we're looking at the response of roots and shoots to light, so we're talking about phototropism. We could also be thinking about gravitropism because they are connected in this way, but this idea that we're thinking about mostly is the fact that it's affecting the growth of the shoots and the root tips. So what group of hormones does that? It's auxins. You can put indoacetic acid in there, IAA, all of those count because they're all correct answers here. State 2 factors the student should have controlled in this investigation. So it needs to be factors that could have affected plant growth or that could have prevented us from being able to compare the results between the groups. Control values are always things that are kept the same. So it's always good to go and check what's already been mentioned in the method that's been kept the same. So each group had 60 seedlings, so the number of seedlings was kept the same. The time was kept the same, although the kind of conditions changed halfway through. Both of this was measured over 48 hours. And so we've got to start thinking, okay, they're all the things that really we should have controlled. So what else could we think about in terms of the factors that could have affected the growth of these shoots that we need to keep the same? So the first one, and one that is always a good go-to, is temperature. Obviously, rate of photosynthesis is going to, and rate of diffusion is going to increase with temperature, so that can affect the growth rate. So we want to make sure the temperature should be the same. Mineral ions can affect growth because obviously they've got to have these mineral ions available in order to grow properly. The volume of water they were given as well to make sure they weren't water stressed. There's loads and you've got more even on the list in the mark scheme, but any 2 of those something that's going to affect the growth of these plants. The start height of the shoots being similar, not necessarily something you have to have, but obviously if you're not going to measure percentage increase from the start size, which they didn't do, then that should be something you should keep the same. So I've gone for temperature and the volume of water. Again, be specific when you're saying water, don't just say water, you should say the volume of water that each set of seedlings was given. For plants in Group A, the students also measured the direction of the growth by counting the number of stems and roots that fell into the following categories. So we've got towards the light, away from the light, and not obviously towards or away from the light, so no direction. So remember, Group A is the one that had the light coming in one direction. It wants us to describe and explain the results in the table. So what patterns can you see? What differences can you see between the stems and the roots and the towards and away from the light? And give some reasons for this. So reasons why we see these differences in the result. So more stems grew towards the light than roots, and more roots grew away from the light than stems. So that's the easiest one because that's an obvious positive phototropic response and a negative phototropic response. So we can explain that using our knowledge of how IAA works. It appears to be that more stems responded to the light than, so in terms of like the number of stems that responded to the light overall, more stems did than roots did. But the fact that there are so many or a high proportion of roots that didn't seem to respond to the light at all could be the result of the fact that we're doing it in unnatural conditions. Could have been some variation in some other factor that could have affected the growth of the roots. It could also be that there was only 24 hours that they were exposed to the light in order to measure the growth. And so that time may not have been enough for the roots to actually have had a phototropic response compared to the shoots. So we have to give a reason for everything as well as describing each row of the table. So I would do it like that systematically. And that's what I've done with my answer. So more stems grew towards the light than roots. This is because of the positive phototropic response. IAA will move to the shaded part of the stem and cause more cell elongation on this side, which causes the shoot tips to bend towards the light source. So I've described and explained for the first row. The opposite was seen in roots, where more roots grew away from the light than shoots. This is because of the negative phototropic response. IAA moves to the shaded side of the roots but inhibits growth on this side, causing the unshaded side cells to elongate more, bending the roots away from the light source. So I've described and explained the second row. There were some shoots that did not show obvious directional growth response to the light. This could have been due to the unnatural conditions of the experiment or the short experiment time, as 24 hours in the light may not have been enough time to see a response in roots. There are two carrier proteins, one which actively transports auxins into cells and another that facilitates their diffusion out of cells. Synthetic auxins have been created which are used as weed killers. They have a shape that allows them to be actively transported into the cell using a carrier protein, but they are not able to use any channel proteins or cross the cell membrane. Explain how these synthetic auxins could cause a plant to die. So we've been told that these auxin can actively be transported in, and then there's a way, normally maybe a channel protein or something, that enables diffusion out. So not necessarily simple diffusion, but facilitated diffusion out of the cell. So we've been told that this synthetic auxin can get in, but it can't leave. And so therefore it's going to accumulate or build up within the cell. If something can get into a cell but can't get out again, then it's obviously going to build up. How does it cause the plant to die? Hopefully we know this about synthetic auxins as weed killers, or weed killers that are containing auxins, then we know how they work. Basically, they cause the plant to grow too quickly and cause it to collapse then. So it grows up too tall without the ability to reinforce those cell walls and get some thickening, maybe lignin and things like that in order to prevent it from falling over. So we need to be able to link those two bits of knowledge together. So we have to say that it can't leave the cells, so it will accumulate. And that's the bit that we've interpreted from given the information in the question. High concentration causes rapid cell elongation, which causes the plant or the stem to grow too quickly and collapse. And then obviously if the majority of your plant is lying down on the floor collapsed, it's not going to be able to be doing photosynthesis as much because it's not standing upright. It's probably going to be shaded by other plants, and that's how it causes it to die. Male pigeons have 2 Z chromosomes, and female pigeons have 1 Z and 1 W chromosome. Feather colour in pigeons is determined by a gene on the Z chromosome that has 3 alleles. The W chromosome has no gene for feather colour. State the type of inheritance demonstrated by feather colour in pigeons. So this is where we get that key phrase where it says the W chromosome has no gene for feather colour, and there is the gene for feather colour is found only on the Z chromosome. So if it's only found on one of the sex chromosomes, then it is a sex-linked gene. Give the genotypes of the following. A homozygous blue male. So this is where we're going to be using our table. So let's break down what it's asked us to give first of all. So homozygous meaning it's going to have 2 alleles that are the same. It's going to be a male, so we know that means it's going to have ZZ, and it needs to be blue in terms of feather colour. So we need to do ZZ, then go to the table to find the blue alleles, and the only one of those 2 options that is homozygous, so 2 alleles the same, is capital B, capital B. So we're going to do ZB, ZB. Remember with sex linkage, you put the allele, kind of the letter superscript above the sex chromosome. Then he wants a brown feathered female. So female is going to be ZW. Brown, there's only one option, which is CC. And this time we just have to remember that there is no allele for feather colour on the W chromosome, so we can just put a C on the Z chromosome, and that's it. Nothing on the W. A pigeon breeder makes a homozygous ash red male with a blue female. Complete the diagram to show the probability of getting an ash red female. So the ash red male is already filled in for us, so then we need to put in the blue female. So ZBW nothing. And then we're going to put the gametes in. So the ash red male can only have ZA gametes, that's all it can produce, whereas the blue female can either have ZB gametes or W gametes. And then I'm just going to move this to give us a little bit more space. So we're going to do our Punnett square. So we're going to do ZA against ZBW. This is hopefully fairly straightforward. And then we just need to fill it in. Same as we would with the normal Punnett square, we're just doing the letters superscript above the sex chromosomes. So we have to have both in there. So ZB, ZA for the 2 top rows, or ZA, ZB, doesn't really matter which way around you do it. And then ZA, ZW for the bottom rows. So that means we've only got 2 possible genotypes of the offspring, either ZA, ZB or ZAW, which means we can only have ash red males is ZA, ZB, that's the phenotype, or ash red females. And just be careful when we're doing sex-linked questions like this, when it comes to phenotypes, you have to put male and female in. You can't just put ash red. You have to put ash red males and ash red females because the sex is part of the phenotype in this case. So the probability of an ash red female then is 50%. Because it's 2 out of 4. So this is hopefully fairly straightforward. It's just, we've got to make sure that we're happy with putting our alleles, kind of superscript of the sex chromosomes, including the sex in our phenotype to make sure we get those marks. Female pigeons are more likely than male pigeons to show a phenotype produced by a recessive allele carried on the Z chromosome. Explain why. So this is just a flipped version of what you'd be asked if you were asked this about humans or mammals. Females only have 1 Z chromosome, and so they can only have 1 allele copy for the genes that are on that Z chromosome, because they won't be able to have the same genes and therefore copies of the alleles on the W chromosome. But males have 2 Z chromosomes, so they can have a dominant and a recessive allele, and then that dominant allele will obviously mask any recessive allele that would give a phenotype. Because females can only have 1 allele for genes on the Z chromosome, but males can have 2 alleles, so can be heterozygous. So this question has to be framed around alleles, not just the chromosomes. So you can't just give the answer that females only have one Z chromosome, because we need to make it about alleles and why they are more likely to have a phenotype from a recessive allele. And it's because they can only have, if they have an allele, they only have one. So if they have a recessive one, they can't then have a dominant one to make up for or kind of give them the other phenotype. So it's got to be about alleles, not chromosomes. Cortisol is a steroid hormone released in response to stress. Cortisol has wide-ranging effects and can influence gene function by binding to and activating the cortisol receptor. The activated cortisol receptor can affect transcription of other genes. Chronic stress can result in prolonged activation of the cortisol receptor, resulting in changes in gene expression of key genes that play a role in cell division and structure. The diagram summarizes the interactions between cortisol, the cortisol receptor, and transcription. Explain why the mode of action of cortisol is different from the mode of action of adrenaline. So we need reasons why they act differently. So hopefully we know that adrenaline is a protein hormone, and so because it's a protein hormone, it works through the secondary messenger model. It's one of those ones like glucagon, for example, that stimulates the secondary messenger system inside cells. Cortisol, it tells us in the question, is a steroid hormone. That means it's lipid-based. That means it can pass through the membrane. And you've got the diagram showing that it's actually binding directly to DNA. That means it's got through the cell membrane and through the nucleus and bound to DNA, much like estrogen, which is another example of a steroid hormone. So this is kind of taking your knowledge about hormones as transcription factors, basically, and also a reminder of what adrenaline does. We've learned about adrenaline in the context of glucose, but we learned that it uses the secondary messenger model, so that is also part of the knowledge that you need for this question. So my first mark is going to come from saying it's a steroid hormone, so it can pass through the lipid bilayers. There's no mark here for just saying it's a steroid hormone, because it tells you that in the question, but the mark is for saying what that means. It means it can pass through the phospholipid bilayer. This explains how it can bind to DNA directly inside the nucleus. Explain why cortisol only causes changes in gene expression in target cells. So we need reasons why there isn't gene expression caused in every cell from cortisol, for example. So why do not all cells respond to this hormone? So it's asking why does it only cause change in gene expression in target cells? If we go back to the question, it talks about changing gene function by binding and activating to the cortisol receptor. And we see that in the diagram as well. So it's the receptor that's only going to be found in target cells, and obviously the cortisol will only be able to bind to its complementary receptor. So only target cells will have the cortisol receptor on the DNA. You could just say this the other way and say not all cells will have the cortisol receptor on the DNA. They say the same thing, that will give you the mark. And cortisol can only bind to its complementary cortisol receptor. A lot of the time with receptor questions, it is about shape. Explain why the cortisol receptor can be described as a transcription factor. Okay, so this is basically what's the definition of a transcription factor. So it's something that changes transcription or affects transcription, and it is nearly always a protein, and it's something that can bind to the DNA. So it's a protein that binds to DNA that changes transcription of genes. So that's what we need to, or changes expression of genes by controlling or regulating transcription. That's enough to get the marks here. It's only a mark, so we've got to give all of that information. So it is a protein bound to DNA, or that binds to DNA, that causes changes in gene expression. Or you can say that regulates gene expression. Scientists wanted to investigate the effects of long-term cortisol exposure on the immune system because previous work has shown chronic stress to cause significant changes in the counts of lymphocytes, phagocytes, and platelets in blood. They used male mice and added rodent cortisol hormone at a concentration of 100 micrograms per milliliter with 1% ethanol to their drinking water for 4 weeks. They had a control group for comparison. Blood was extracted before and after the cortisol treatment, and the number of T cells, B cells, and phagocytes was counted using fluorescence-activated cell sorting, or FACS. This is where the fluorescent tag antibodies specific for each cell type are added to the blood, and a laser beam is used to sort the cells. Analysis of the DNA in each cell was also carried out to check for methylation in genes known to be affected by cortisol. The results are shown in the graphs, in the two graphs. I'm only going to show one here just because of space. We'll look at the graphs when we need them on the same page. Suggest how the scientists treated the control group of mice. So suggest questions are always your own knowledge plus information from the question. This idea of like, how would you treat a control group when you're being told the treatment group is very common for like clinical trials and medical tests. Basically, they want to know what they did to the control group to keep it as a control so it can be a comparison. It says for comparison in the text. And they told you that the treatment was that they added rodent cortisol hormone at a certain concentration with 1% ethanol to their drinking water for 4 weeks. So the control group should have been exactly the same, but just without the cortisol. So they still get something else like water or nothing, and then the 1% ethanol to the drinking water for 4 weeks. So it should be the same. We should include the 1% ethanol because that's not cortisol, and just to make sure that that's not having any effect. But always need to say treat them exactly the same or give them exactly the same treatment, but with no active ingredient or whatever it is that's causing the change. In this case, it's the cortisol. So if this was a human trial and they were doing an injection of some kind, a vaccine or something, you would still have to inject all of the other people with saline or whatever solution the vaccine was in, just without the actual vaccine in it. And that is showing that it's exactly the same, but without the active ingredient. Explain why the fluorescently tagged antibodies would have been able to identify the different types of white blood cells. So it wants reasons why the different antibodies we've got are going to be able to identify or be able to be used to identify the different types of cells. It said they're specific for each cell type in the question. So we should know what that means. That means that they are specific, which means they're only going to bind to one of the cell types. And then we just need the reason why they're only going to be able to bind to one of the cell types each, and that'll be due to their tertiary structure. Again, we've got this idea of complementary shapes fitting together. It's like a recurrent theme throughout all of this biology, and if you've anything to do with specificity or something only binding to something, it's very, very commonly going to be about sort of shapes fitting together. So then we just have to relate that specifically to an antibody this time. So we should hopefully know that their variable region where the specific tertiary structure is, that's the part that's going to bind. And obviously we know that they bind to antigens. So that's what we're going to be saying. They're going to be specific antigens or the different types of cells or specific proteins, receptors that these antibodies are going to bind to. The tertiary structure of the variable region is a specific shape that is only complementary to the antigens on each type of cell. This means it will only bind to one type of cell. Using the information in the graph, explain why people are often more susceptible to viruses when suffering from long-term stress. So there's information in the graph that we can get clues from or talk about, and we need to explain why, so give reasons why people are more susceptible to viruses. So they're going to have a weakened immune system or a weaker immune system. They're going to be less likely to be able to fight the viruses off or destroy them because they're suffering from long-term stress. We need to link these two things together. So link to cortisol, link to the white blood cells that we've got in the graph. What has happened to the white blood cells? How can we kind of link that to this idea of a weaker immune system or not having the ability to fight off a virus? So we can see on the graph clearly the number of T lymphocytes and B lymphocytes decreased, and there's no overlap between those error bars. So that's a significant decrease for both of them with cortisol being present or high levels of cortisol being present. How can we link that to needing to be able to destroy or get rid of a virus? Well, you need your T and B cells to produce antibodies. You need the T cells to activate the B cells, then the B cells to divide in order to produce plasma cells to make the antibodies. And that's how we fight viruses that are inside cells. So this is why having more phagocytes doesn't actually help us, because phagocytes aren't necessarily going to be able to destroy viruses because they'll be replicating inside affected cells. And we need antibodies really to actually fight a virus. it's going to be very difficult for your body to fight off a virus if you just have phagocytes working. So if there's no activation, there's no mitosis, there's no plasma cells being produced, that's what's going to cause this weaker immune system, because we're not going to be able to produce as many antibodies. So from the graph, we can give our first point, which is that cortisol being high significantly reduces the number of B and T lymphocytes in the blood. I could have talked about the error bars here, not overlapping, but I didn't. I just went straight on to explaining what that meant, but you could get a mark for that as well. I did put significantly, and I know that because of the no overlap. Fewer T cells means fewer B cells will be activated, so there'll be fewer plasma cells produced by mitosis, so fewer antibodies to fight off the virus. And I've literally just said, this won't happen, so this won't happen, so this won't happen. And that's fine, because that logically makes sense to kind of do it in that order. There's some other marks available. If you spoke about T cells being less likely to come across an antigen-presenting cell or the pathogen itself and be activated, same for B cells. You can also put that, because that is true. If there's fewer of them circulating around in the blood, there's less chance of that happening as well. But yeah, there's plenty of marks there. But just as long as we know how our immune system functions in order to produce those antibodies, and we can see why having fewer white blood cells, specifically T and B cells, is going to make us more susceptible to a virus. Cortisol is known to affect cell division and apoptosis. Using graph B, explain how the changes in DNA methylation caused by cortisol exposure could have caused the changes in the number of lymphocytes. Okay, so we've clearly got a decrease in methylation in both T cells and B cells with the cortisol. So this is an epigenetic question. We should hopefully know that decreasing methylation is going to increase transcription of those genes, because it's going to loosen them from the histones. They're going to be more easily transcribed by RNA polymerase. That means that's going to activate transcription, increase transcription, so more protein of whatever gene it is going to be made. We know there were fewer cells, right? There were fewer T cells and B cells from graph A. I haven't put it on the slide, but from graph A you can see we've got a decrease in those cells, and we just talked about that in the previous question. So that means less cell division, or reduce in cell division, or an increase in cell death. So apoptosis is cell death. So either you can pick either one of these or both, but the proteins that are being increased in transcription, which is leading to this more protein, has got to be linked to these, because this is then going to be the thing that's going to cause that reduction in T cells and B cells, either an increase in cell death or a decrease in the rate of cell division or mitosis. So DNA methylation is decreased in T and B cells when cortisol is high. This means transcription of genes is increased or activated. So that's one mark there for describing what I understand is happening with the methylation. More of a protein that prevents mitosis or causes cell death must be made, which reduces the number of T and B cells. So I've written both. You probably should have just picked one. So either pick about preventing mitosis or causing an increased rate of cell death. One of those two must be being made, and then link that back to the reduction in the number of T and B cells. You've already talked about that, but it just says cause the change in the number of lymphocytes. It doesn't say decrease. So we've got to kind of link that into our second point. So this question was hard. It was designed to be hard. It's a paper 3 question that jumps all over the place with a bit of epigenetics, a bit of transcription factors, a bit of secondary messenger model, a bit of how hormones work as transcription factors, a bit of immunity, a sort of question about data and control in experiments. So there's lots of stuff in this question, which is very typical of a paper 3 question. You've got to be able to read these questions and kind of be able to think about, okay, what bit of knowledge do I need for this? And it's not always going to be just because it looks like a transcription factor question. That's not what the whole question is going to be about.
Okay, so we're going to look at the 2 essay questions in a minute. I'm going to plan both and then write both and go through my essays that I write. The main thing I want to do before that is to focus on this assessment guidance. So This is really helpful. It's from the mark scheme. If we're aiming for this high bracket, so the section where we're going to try and get over 20 marks, then we need to have an essay that flows and is interlinked with points that all contribute to the theme of the question. There need to be clear links between at least 4 topics, which is a minimum. The biology needs to be detailed and comprehensive A-level content beyond GCSE level. There needs to be no significant errors in that biological content and no irrelevant material that do not link together to the question. And if you want to get 24 or 25 marks, then you need to have evidence of wider reading by adding information that is relevant to the question, but not required by the specification. So not one of the bullet points or incorporated into the specification from AQA. That means it can be from another exam board and still be A-level content. That's fine. It must be at least 3-4 lines and not just single sentence facts. So this is all the things I've been thinking about when I'm planning and writing my essay. If I'm going to choose which essay I'm going to write, can I confidently cover at least 4 topics at a minimum? And for all of those topics, can I give the same level of detail, A-level content that I know without making any significant errors? And does everything I'm going to say or that I know about that topic, can I relate it to the question? Once you've done that, you can start, and you're only going to be able to do that by reading the question and listing, bullet pointing some topics that you think you're going to include. Okay? And then once you've done that, you can decide, okay, can I talk about all of those 4 to 5 things in lots of detail? Can I bring any other points that link into it, that link to the question? And is there anything beyond the spec that I know that I can link in here? Okay, so that's where we're going with this. Obviously, it's still quite sort of easy, I would say, to get into that bracket where it's about 16 to 20 marks. I would say that's probably the average mark base that people are trying to aim for, rather than trying to get into this over 20 mark. I would say you've got some points that are detailed, but there may be some which are less well developed. So this is where you can end up spreading yourself a bit too thin and either mentioning loads of things and not going into much detail and not developing that really high level A-level content in there and just superficially explaining them and not actually going into much detail. And you are allowed like one significant error, so something fundamental that's wrong with your biological knowledge, or, and there's a big point there, or one irrelevant topic which detracts from the quality of the answer. So you can either have one significant error where you really don't sound respiration or something, or you can have one topic where you talk about a topic for a while but it's not relevant. And that will still allow you to get into these brackets, but it may get you a kind of lower to medium answer. And that's what I would say. Try not to spread yourself too thin with too many topics in superficial detail, and try not to make sure that all of your topics that you are going to pick are going to relate to the question.
So our first essay question is the importance of variation and diversity in organisms. So my first thought when I see variation is genetic and environmental, the 2 different types of variation. My first thought when I see diversity is biodiversity and then maybe genetic diversity. So these are kind of like my 4 points, main points that are kind of that sprung to mind that think about what can I expand on these, kind of give myself some actual topics in detail to talk about. So biodiversity, think about ecosystem stability. So yeah, thinking about why it's good to have a biodiverse ecosystem. For genetic diversity, I'm thinking about things like, okay, if you have high genetic diversity, then you're more likely to survive as a population because your ability to be able to cope with environmental change is higher. You're not going to be susceptible to diseases, or a change in the environment can't just wipe out your whole population. For genetic variation, we know that there's mutation, obviously, but also genetic variation is produced by meiosis. So I can do a nice thing about talking about how that works and happens in meiosis. Environmental, I'm thinking obviously there's environmental variation, but my thinking goes to epigenetics, partly because I really love epigenetics and it's one of my favourite topics, but also I think it's a good place to potentially get some extra knowledge from the spec in, or just to have something different to talk about. Obviously with genetic diversity as well, we're going to mention natural selection. You can't answer this question without mentioning natural selection really, so that's another nice kind of process. This wouldn't be my choice of essay out of the 2, because I always prefer, and I think it's a lot easier to go for the kind of more specific one where there's lots of processes involved, some biochemistry and the kind of cellular level stuff, because that involves a lot of process, a lot of detail that you can remember, and I find that a bit easier. But if you can get things like natural selection, which is a nice process, meiosis, which is a nice process that you can have learned the detail of quite nicely for other parts of the papers in paper 2 and paper 3, then it's nice to be able to put those in where you can when you get one of these kind of broad questions like this. So now that's a brief plan. That is not enough. That is what I would write. The pink stuff is what I would write in a brain dump when I was first choosing which question I'd do, and then from that brain dump I would decide. So if I've decided to do this essay, now I need to turn that into a bit more of a detailed plan. So for meiosis, I'm going to definitely talk about random fertilization, crossing over, and independent assortment. Then I have to make sure, always, I'm linking my paragraphs back to the question in terms of importance. So why are these 3 things important? Well, obviously, they increase genetic variation, they create unique organisms, the mixture of alleles you get from your parents determines your phenotype, and that's obviously going to determine everything about you and everything about the organism. So that is kind of where I'm going with that, because what we don't want to do is have our importance be the same for each paragraph, which it could be. If you've got really kind of down to the nitty gritty, you're basically saying variation is a good thing, more variation is better, more chance of survival, but you don't want that to be your repeating importance. It's got to be a bit more specific than that. So then we've got mutation, genetic mutation. That is obviously a change to the DNA-based sequence, so we can mention some examples like substitution, deletion, addition. The fact that deletion and addition cause a frame shift, and that the importance of mutation is that ultimately it changes protein structure and can therefore affect phenotype. So if I think about that, I could probably talk a bit about protein synthesis. Not like really super level detail as if we were doing the protein question, because we're going to do it for that as well probably, but at least I can talk about protein synthesis as another process, another A-level detail that I can get in there, and that links to this idea. It's going to help me explain how it's causing the change in the primary structure of proteins. And obviously they can be beneficial or they can be negative. If they're beneficial, then that can link nicely into us talking about natural selection. So environmental variation, I'm going to mention epigenetics as well. So this is about changing gene expression. So it's another way of creating variation. The thing about epigenetics is that it allows organisms to cope with changes in the environment, but in a much more rapid and kind of individual way than, say, the process of natural selection over many generations and becoming adapted to an environment. It allows you to pass on heritable changes in gene expression, not DNA, but just the expression of genes to your offspring in order to prepare them for the environment they may be being born into, for example. And yeah, it just allows them to increase their chance of survival. So a paragraph about natural selection and speciation, probably, because natural selection does come up at GCSE. One of the main things I want to try and make sure that we avoid in this essay is slipping very much into GCSE topics, because with the exception of things like meiosis in detail, and some things about kind of the hardcore genetics and epigenetics, we could slip into very easily GCSE tropes here and talk about things at a very surface GCSE level, because we would have done all of this, and it's about making sure we're including all that A level and specifically try and getting the A2 content in where we can. So speciation needs variation. Okay, so this whole thing that we can link this into is that the variation that we see created from meiosis and things like that, and genetic variation, is needed for this process to happen. It allows change, it creates these organisms that are adapted to their environment, and that's its importance, isn't it? It's variation allowing organisms to cope with environmental change, survive, reproduce, and end up becoming very adapted to the environment they live in, and evolution to happen, which we wouldn't have if we didn't have variation in the first place. So that's a nice kind of importance link there. Biodiversity and ecosystem stability then. So you'll notice I've got to push this to 5 topics. Not saying don't do 5 in the exam and just do 4 at all. If you are unsure, especially with one of these kind of maybe broad ones where you're not really sure whether everything you've included is very easy for you to link to the importance, or you just want to try and cover a bit more bases and you think that you have time. So I put this one at the last point because I would say this would be my weakest paragraph. So I'm putting it last because if I don't get time to do it, it's not the end of the world. And if I get time to do it, I know it's not going to be strong anyway, whether or not I have time. And so therefore I'm putting it at the end. And sometimes it's worth doing that. Just think about the order of your paragraph sometimes. If you've got one that you know you're weakest on, or you've added in their fifth paragraph to try and kind of cover a bit more bases, that's absolutely fine as long as you're putting the detail into all the other paragraphs and you make sure that you're not kind of like skipping through each single one to try and cram 5 in. If you can do 4 detailed paragraphs and try and add a 5th, then that's fine. But don't put your best subject with your best subject knowledge at the end. So I'm going to talk about high biodiversity created by variation means a more stable ecosystem, and that links into interdependence. So obviously all organisms relying on other organisms for things like food, shelter, pollination, things like that. So the more diversity and biodiversity there is in an ecosystem, the more likely they're going to be able to support each other. So if something happens, if 1 or 2 organisms die, disappear, or something happens to them, the whole ecosystem isn't going to collapse because everyone's got multiple options for food, habitat, shelter, etc. And so they'll be able to cope and survive. We've got some long-term examples of stable ecosystems before human activity and climate change started to ruin them. Things like rainforests and coral reefs and stuff like that. So the importance is that obviously without biodiversity, ecosystems would collapse and we'd get extinction, which we are seeing now in places where human activity is causing that biodiversity to decrease. Okay, so this is my first paragraph. What I'll do is I'll read through the paragraph and then I'm going to point out where I linked it back to the question in terms of importance and sort of point out where my focus is on variation. And if at any of the points through the paragraphs I've used what I think is kind of beyond the spec knowledge as well, I'll point that out. So genetic variation is produced through sexual reproduction and the formation of gametes by meiosis. This process determines the alleles an organism inherits and therefore what versions of proteins they will be able to produce that will ultimately determine their phenotype. Meiosis produces gametes that only contain half the genetic information from each parent. The fertilization of 2 random haploid gametes restores the full number of chromosomes to make a diploid cell. This will contain a random mix of half the chromosomes from each parent. The process of meiosis also increases genetic variation by creating different combinations of parental chromosomes and alleles in each gamete, making them genetically unique. In prophase of meiosis 1, homologous chromosomes pair up to form bivalents. Some will cross over the arms of non-sister chromatids and a chiasma forms, where the DNA breaks and the sections reattach, they bind to the opposite chromatid. Genetic material has been exchanged, which creates new combinations of alleles on chromosomes that are different to the parental genotype. The separation of alleles in this way is more likely to occur between non-linked genes on autosomal chromosomes, as their loci are likely to be further apart on the chromosome. In metaphase of meiosis 1, the pairs of homologous chromosomes randomly line up along the equator of the cell, and are independently assorted when they are then pulled apart to the opposite poles of the cell by contracting centrioles during anaphase. This determines which chromosome of the pair will end up in each gamete. There are 2 to the power 23 varieties of gametes that can be produced in humans, which massively increases the genetic variation further. The inheritance of the unique combination of chromosomes and their alleles is what makes no 2 organisms genetically identical, with the exception of identical twins, and is the basis of the majority of variation in organisms. The expression of these alleles will then determine the phenotype of the organism by coding for specific proteins. The control of gene expression can introduce further opportunities to create variation. So, I start with a bit of importance. Okay, so a bit of kind of thinking about why I might want to say this is important. So talking about determines the alleles they'll be able to produce, which ultimately determines phenotype. Then I go into lots of detail, A-level detail that's all correct about the process of meiosis and how it creates genetic variation. And then at the end, kind of like signposting throughout the links to variation. So what I talk about variation that I'm mentioning, increasing genetic variation, to make sure that it's showing that it's relevant to the question. And then I end with another little bit of importance. So sometimes I like to bookend it, a bit at the front of the paragraph, bit at the end of the paragraph, just in case I forget. So if you put a bit of importance at the front of the paragraph and you forget, or what you put at the end of the paragraph isn't enough, you've got a bit at the start as well. And so the importance link was obviously about how it's going to determine the phenotype of an organism and their expression of those genes. For each gene, mRNA is transcribed from the DNA in the nucleus using the enzyme RNA polymerase, which joins complementary free RNA nucleotides that have aligned with their base pair next to the template DNA strand. After non-coding introns are then spliced out of the pre-mRNA and the coding exons joined together in eukaryotes. The mRNA leaves the nucleus and binds to a ribosome to be translated. Coding exons are arranged together in the correct order that will produce the correct sequence of amino acids of the protein coded for by the transcribed gene. Without this correct stage, the correct protein would not be formed or would not function correctly. Ribosomes are made of a large subunit and a small subunit which fit either side of the mRNA strand. The structure of the large ribosome subunit contains space for 2 tRNA molecules to bind to their complementary anticodon on the mRNA strand. The ribosome then acts like an enzyme, as it catalyzes the condensation reaction forming a peptide bond between the 2 amino acids on the tRNA molecules to start the formation of a polypeptide chain. The proteins made from genes alter the structure and function of cells, and the combination of genes expressed in each cell causes them to become specialized to carry out specific roles in organisms. The genes expressed in the cell of an organism will affect their appearance and function. Inheritance of recessive alleles or mutations that occur during DNA replication or transcription can produce non-functioning proteins that lead to disorders. Mutations are changes to the DNA base sequence that occur as errors when copying the genetic information. They can cause a change to the order of amino acids used to make a protein, which can affect the protein's structure and function. Many mutations are silent or have no effect on the phenotype because they do not cause a change in the amino acid used. This is due to the degenerate nature of the DNA code, where multiple different codons can code for the same amino acid. Deletion and insertion or addition mutations change the number of bases in the DNA molecule, which causes a frame shift. This means that every codon after the mutation will be changed, and it can have a large effect on the protein produced, as nearly all amino acids after the mutation will be different or in a different order. These mutations are likely to cause changes in the phenotype, and they can be negative or positive. Sometimes these mutations can offer an advantage to an organism, which can lead to increased survival when their environment changes. This occurs through the process of natural selection. Okay, so long paragraph. I probably included more detail about transcription and translation than maybe I needed to, and actually you could probably cut that down because you could potentially see that as irrelevant information. Some of that detail is probably unnecessary, but this is the kind of level of detail we need to be thinking about when we are writing these essays. So I've got talk about how genes are expressed. Realistically, the process of transcription and translation is about how genes are expressed. So that process of how genes are expressed is valid, because then we talk about mutations, which are a form of genetic variation, which changes the expression of the genes potentially because it changes the protein that's being produced. So as long as you can kind of try and link this in together, it does make sense. Just be clear that that bit about protein synthesis, you're linking directly to this idea that that is the expression of genes. That's how genes are expressed. And then we link to mutation, which makes sense because we're talking about how that can cause different proteins to be made, which is a form of genetic variation. So all the pink highlighted parts show how this flags the importance of these processes. So the importance of transcription and splicing, the importance of translation, the importance of mutations is kind of highlighted with each of the pink sections with some detail about each process in between. The one thing I would say is lacking in this paragraph is link to variation. I don't know if I mentioned the word variation at any point in this paragraph, and in hindsight I would have gone back and maybe thought about making it clear why and how this links to variation or genetic diversity. And that's the things we're lacking in this paragraph. But it's got A-level detail in, and it is showing the importance of these parts of genetic variation to the organisms. Natural selection is a process through which organisms change over time and is one process that drives evolution. It relies on variation in a population and often random genetic mutation, which creates a new advantageous allele. Selection pressures such as competition or temperature will cause a struggle for survival where not all organisms will be able to survive and reproduce. Organisms with variations that make them more likely to survive have a selective advantage. They are more likely to reproduce and pass on those alleles to the next generation, so these favourable alleles increase in frequency in the gene pool. If selection pressures are different for different populations that are isolated in different geographic locations, allopatric, or if individuals in the same habitat become reproductively isolated from each other, sympatric, then speciation can occur, and new species can emerge once the populations or individuals can no longer interbreed to produce fertile offspring. An example of allopatric speciation is Darwin's finches, which are all descended from an ancestral species of finch from mainland South America. The population of ancestral finches became geographically isolated from each other on the different islands of the Galapagos. On each island, there was different food sources which acted as a selection pressure. Birds with specific beak shapes on each island were more successful at feeding, and therefore more of them survived to reproduce and pass on alleles which produce this beak phenotype to their offspring. Over time, the frequency of the alleles increased in the population on each island, and over many generations, the phenotype frequencies changed. As there were no interbreeding between populations from each island, the gene pools were separated, and eventually the birds became reproductively isolated from each other and can no longer produce fertile offspring, making them distinct species. There is now great evidence of adaptive radiation in the birds of the Galapagos that show great variation in phenotypes due to the different niches occupied on each island. Without initial variation in the original ancestral population, these new species would not have arisen through this process. Organisms that carry out asexual reproduction have large populations of genetically identical individuals. This makes them more susceptible to becoming extinct due to environmental changes, as there are fewer alleles in the population, which could allow some individuals to survive the changes, for example, a new pathogen. Okay, so that last bit I've linked to importance there. This could have been an importance link back with meiosis, potentially, because obviously that's how sexual reproduction occurs. But again, there we were talking more about how it creates the genetic individual genotypes and phenotypes, which creates individuality in organisms. And so we can put this in here to kind of give you the idea that if you don't have variation because you do asexual reproduction, then you're not going to have the opportunity to potentially be able to cope with change because you can't go through that process of natural selection because there isn't any variation. And there's fewer opportunities or chances that any alleles that you already have are going to offer that ability to survive if something changes. So this is a paragraph that's basically just natural selection and speciation. So sort of pushing that idea of natural selection a bit further with speciation to make it definitely A-level level. So there's lots of detail. There's a full example about Darwin's finches. You don't have to have an example, but that allows for use of that language and the steps that occur in natural selection without just listing sort of what would have been in a few bullet points. And it sort of shows extra knowledge, a little bit beyond the spec, because the actual Darwin's finches example isn't on the specification. So learning that and having that to hand, even with a bit more detail in terms of maybe examples of the different food types on the different islands potentially, could also help there. But it's just showing that kind of deep knowledge of natural selection and then eventually the process of speciation and why that's an advantage. So again, sort of starting with importance, like the fact that natural selection is a process that changes over time. It's a process that drives evolution. That's really important. And then every time I've highlighted in purple here, every time I kind of mention the idea of genetic diversity, so mutation counts that, or variation, which was many times actually mentioning that word. And then at the end, especially linking that idea that without that initial variation, we wouldn't have had that new species being produced. And then that links straight into that nice little bit of importance at the end about why it's important to be able to have this variation produced through sexual reproduction. There was lots of detail in here with lots of keywords, key language, again, all correct and all A-level level. As well as genetics and selection pressures, the environment can also play a more direct role in shaping an organism's phenotype. As well as phenotypic changes that are the result of injury, for example scars, lost limbs, or disease, for example reducing lung function after an infection, the environment an organism develops in can have an impact despite their genetic potential by affecting gene expression and therefore what proteins are produced. For example, if an individual has inherited alleles from their parents that could allow them to grow very tall, but during childhood they suffer from a poor diet, where they do not receive sufficient nutrition, including enough protein, they will not be able to grow at the same rate as others who may have similar alleles. This poor diet results in the individual not sharing a similar phenotype, being tall, with their parents, despite sharing the same alleles as them, due to an environmental factor during their childhood development. The environment can also cause heritable changes to gene expression through the process of epigenetics, which results in phenotypic changes that are not caused by changes to the DNA base sequence. In eukaryotes, DNA is held in the nucleus surrounding proteins called histones, which bind to the DNA and help the DNA to condense into chromatin. Histones are covered in chemical tags that determine the shape of histone DNA complexes, and they help to control the transcription of genes. Increasing the methylation of DNA, by adding a methyl group to DNA, prevents transcription factors and enzymes from binding to it, which reduces its expression. DNA methyltransferase enzymes attach the methyl group to the cytosine bases on the DNA molecule. These interact with the histones, binding DNA more tightly. Another mode of epigenetic control is acetylation of histones. Adding acetyl groups using acetyltransferase enzymes loosens DNA from the histones, making it less condensed. This increases gene expression by allowing RNA polymerase access for transcription. When acetyl groups are removed from the histones by histone deacetylase enzymes, the chromatin condenses and transcription factors and enzymes cannot access the DNA, so it is not transcribed. These epigenetic changes, through control of acetylation, deacetylation, and changes in methylation, have been shown to be caused by many environmental factors, including availability of food, social interactions, disease, and chemical exposure. An example is drought resistance in plants. Parent plants experiencing drought have demonstrated epigenetic changes to alter expression of genes to reduce water loss. Seeds germinated and grown from these parent plants were also shown to have the same epigenetic differences despite them not experiencing drought. It is thought that these changes are passed on to offspring, assuming they would experience similar environmental conditions, and therefore this prepares them for survival. Faster than adaptations produced through generations of natural selection, the ability to inherit epigenetic changes is important, as it allows the inheritance of behaviours which will make offspring automatically prepared to deal with the environmental changes they may experience. So again, briefly started with kind of an importance, so the environment playing a role in shaping an organism's phenotype, so this is kind of producing environmental variation. Probably again could have mentioned environmental variation a little bit more at the top, but as long as you're kind of demonstrating that you understand when you say things like phenotypic changes, phenotypic differences, changes to gene expression, that is variation because you're suggesting that these are different in these different organisms. Or, for example, the section about being different to their parents. So despite having the same alleles, they are different. That is linking and suggesting of variation. Then there is a lot of detail about epigenetics. It is one of my favourite topics, I won't lie. So this is something that I know, and this is extra information from beyond the specification. It's not just a few words or a sentence of fact. It is explaining, it's got the enzymes in, it talks about both types of acetylation, whereas normally in the specification you've just looked at one type of acetylation of histones, not deacetylation. It adds in the enzymes that carry out that epigenetic change. It also includes an example of epigenetic changes in plants, which again shows an understanding and shows that highlights the importance of this. So the whole sort of last few sentences about the importance of epigenetics being able to allow an organism to adapt faster than generations of natural selection, but make sure that your offspring are able to cope and survive, more likely to survive in the environment they are being grown into, is obviously a really important thing that helps organisms to survive. So there's a nice importance link there. Final paragraph then, so this is my, as you can see, shorter paragraph, which is the biodiversity paragraph. The functioning of many ecosystems relies on the biodiversity or number of different species in its community. The most long-term stable habitats have the highest biodiversity and have been able to survive many environmental changes. Examples include the South American rainforests, Australian coral reefs, and the European ancient woodlands. Now, human activity and climate change is greatly affecting global biodiversity by destroying habitats or changes in abiotic conditions. We can see the consequences in increases in species extinctions. The more biodiverse an ecosystem, the more sources of food, habitats, and niches there are for organisms to occupy, the more stable an ecosystem becomes, as interdependence between species increases. This means there are multiple resources for each organism to depend upon, so the reduction in one does not drastically affect the population. Once the carrying capacity of an ecosystem has been reached, there will be small fluctuations in population numbers, for example, predator-prey cycles, but the population cycle size will remain relatively stable. This creates a balance which allows long-term stability of the ecosystem. Ecosystems with low biodiversity are more at risk from environmental change, where small biotic or abiotic changes can greatly affect the number of individuals in a population that will have a great impact on other species in the community, which rely on it as a source of food or habitat. It can increase competition or predation among remaining organisms, which reduces their populations further and puts the community at risk of extinction. Greater genetic variation or genetic diversity in populations will enable them to withstand environmental change and avoid extinction by enabling them to adapt to their new circumstances through the process of natural selection. So again, opening with kind of an important statement. So function of ecosystems relies on biodiversity to be high and explaining why that is. Ending again with a link back to variation. So if all of this stuff I've just talked about, if you have greater genetic variation or greater genetic diversity in populations, they're going to be able to withstand environmental change. So that's kind of a recurring theme that's come back. But the majority of the chunk of this paragraph is detail, but it's not just facts. It's not just a process. This is a section chunk of detail that explains why high biodiversity is good and important for ecosystems. So this in itself is a whole sort of paragraph of importance with some detail, with some key language and key words in it, because it's not just a statement of facts about what biodiversity is. It's nice and sort of linking and holistic paragraph there. So it's still good, even though it's on the end and it's a bit shorter, because it's lots of explained detail as opposed to just process detail. Okay, so as you know, there's a generic mark scheme for the enzyme, but we always try to link some related content ideas and some specification points that could be relevant to this. It's not an exclusive list though. There are other things you could potentially include as long as you can relate them to the question. So I'm just going to highlight all the things we talked about in that essay, which shows that it was very easy, despite us thinking we had kind of 4 or 5 topics, that actually we've covered quite a few spec points. So genetic differences due to mutation, meiosis and how it causes genetic variation, advantages of genetic variation, natural selection evolution, speciation, the 2 types, genes, alleles, proteins, gene pools and frequencies, stability of ecosystems through biodiversity. Loads there, way more than sort of 4 topics covered. So as I said, make sure you've got lots of detail that's at A level. It's very easy to slip into GCSE language here. A lot of this stuff, with the exception of kind of the hardcore genetics, things like mitosis or asexual and sexual reproduction, natural selection, are some things that could very easily, and even just like biodiversity, are some things you could easily talk about at GCSE surface level. So make sure you're including all of that A level detail where you can. Really check back over for any silly mistakes with language. So things like saying organisms adapt, they adapt, and using it in that way to try and say that like they choose to adapt, or it's their kind of behavior, they decide to adapt, that kind of thing. Homologous chromosomes, making sure if you are going to do meiosis that your description of meiosis and what happens in crossing over and independent assortment is on point and you are not making any silly mistakes that show that you don't understand the actual process of meiosis, because any major errors in scientific knowledge will kind of drop us down in the mark scheme. But yeah, hopefully you've written a lovely essay and included a lot of this stuff. Okay, on to our second essay, the easier of the 2 options, in my opinion. The importance of proteins and their structure in the control of processes in organisms. 3 elements to this, but there's so much that you can write about for this. So much. And it's a lot of processes, and there's loads of chance of getting A-level detail in here. There's so much. So this was kind of my plan, which concluded a lot, and I obviously had to cut it down and pick 4. And there'll be even more on the list of things we can look at the end as well. So first of all, think of some proteins, enzymes, antibodies, hemoglobin, receptors, carrier proteins and other membrane proteins, which could link back to receptors as well, or transport proteins, transcription factors, hormones, specifically obviously adrenaline and glucagon, the protein hormones that we've learned about. So many already, too many to talk about all of those anyway. But that's a good start, because once you've thought about some proteins, then that's going to make you think about some processes. And once you've decided to match them up, that's when you can decide what topics you're going to do. It mentions structure in the question. That means that for all the proteins, not necessarily for all the proteins, because we're going to mention a lot, But for the proteins we're going to look at, we should focus on how their shape or structure helps them carry out their function. So at one point, or at least somewhere in our essay, we need to mention the primary, secondary, tertiary, quaternary structure of the proteins and potentially bonding as well. That should be a focus. We're not just saying, this protein does this, move on. This protein does this, move on, because there is a structural element to the question. And it's about the control of processes. So let's think of some processes that we can now link to these proteins that we thought of. So respiration, obviously, photosynthesis, obviously, immune response because of antibodies, DNA replication, transcription, translation because of the enzymes, but also transcription factors can be in there. Membrane transport, so co-transport, active transport, anything that's talking about transport, so any like gated ion channels, anything like that. Digestion, obviously a big one with enzymes. There's loads there. Oh, also hemoglobin, oxygen transport. And then hormones, don't forget those. So blood glucose control, or the fight or flight response for adrenaline, or blood glucose control for adrenaline as well. But also that links into the secondary messenger model. So yes, that can be talked about as well because that's a process. So I'm going to pick 4 of these. I'm going to try and pick a range. So I've gone for respiration, which is obviously kind of a biochemical process. I've gone for the immune response, that's kind of a whole body response. And I like talking about antibodies. That'll be a nice one for structure. DNA replication, transcription, and translation, a big one. Lots of enzymes you can talk about in there. And then oxygen transport, because I can talk about hemoglobin. And my thinking behind this is I can add some extra beyond the spec information for hemoglobin, which I know is an easy fit. So as I said back when we looked at the mark scheme, there are points to get you into that top 24 or 25 marks for extra specification content. I'm putting it in these essays to demonstrate to you how it can be done and the level it needs to be done to, but I really would not be focusing on that as my main priority when planning these essays and revising for these essays. If you can get way higher than average by getting 23 out of 25 for the essay for writing an excellently detailed, accurate, correct, A-level detail, synoptic essay where everything is related back to the question, then that is amazing. And that's what you should be aiming for before you even start thinking about adding that extra beyond the spec information, unless you have it to hand, unless you know it really well, and you know that it's something that you can just slot in because it's something you read in a textbook somewhere else from a different exam board or something. I would not be focusing on that as part of the plan when you are doing these essays in the real exam. I really wouldn't. I'd be much happier for everyone to just get 23 out of 25 for an amazing essay that was really well thought out and really well planned. Okay, so I've got first topic respiration, then I'm going to list the proteins that I'm going to talk about. ATP synthase, the electron transport chain, NAD and FAD, because I'm going to go through the process of making ATP, and they're all the proteins I want to talk about. The importance of respiration is obviously the production of ATP for energy for living processes. Nice and easy. Immunity. I'm going to mention antibodies, I'm going to mention receptors, and I'm going to mention antigens. They are all proteins, and they're all relevant, and they'll be easy to talk about in the process of immunity. The importance of immunity, again, a nice easy one. Defends the body, creates immunity to pathogens, helps us survive. Transcription and translation, RNA polymerase and ribosomes. Both proteins, both ribosomes made of protein, as well as other things, but both proteins and both requirements for that. And obviously, that's a big one because you're talking about the importance of that is that you're making proteins, which is relevant to all the other proteins you've talked about. It's about how genes are expressed, which is what we've obviously been talking a lot about in the previous essay. And yeah, there's loads you can say here. Oxygen transport, hemoglobin. The importance, obviously, is because of gas exchange or gas transport even for respiration. If we don't transport oxygen to cells, they can't do respiration, which means they can't produce ATP, which means we don't have any energy. So there's a nice one there, but also excretion, like you need to remove the carbon dioxide as well, which is where my beyond the spec knowledge comes in. I'm going to talk about the enzyme carbonic anhydrase, which is a protein, and how that allows the shape change and the kind of dissociation of oxygen, part of the Bohr shift, but it's kind of an extra bit of detail that AQA specification doesn't include, but OCR specification does. So it's something that's on there that's an extra then. It's not part of the specification, but it's a bit of more detail about explaining how oxygen dissociation happens at respiring tissues. One of the main ways that processes are controlled in an organism is through the expression of genes. What genes are expressed or not in a cell can greatly influence the cell structure and function and leads to specialization of cells for specific roles in tissues and organs. Gene expression occurs through the process of transcription and translation, which are regulated by many proteins. DNA transcription requires the enzyme DNA helicase to break the hydrogen bonds between bases to expose a template strand of DNA. Then RNA polymerase joins free complementary RNA nucleotides that align to the DNA bases. The specific tertiary structure of the RNA polymerase is created by the specific sequence of amino acids in its primary structure, which determines the position of the R groups. And this will determine the position of the ionic and hydrogen bonds which create the specific tertiary structure, which forms the shape of the active site, which works by reducing the activation energy of the reaction. In this case, RNA polymerase can bind to RNA nucleotides to catalyze the condensation reaction to form a phosphodiester bond between the ribose sugar and phosphate group of each nucleotide. This forms a strand of pre-mRNA. Enzymes are required to splice the mRNA to remove the non-coding introns so that the coding exons are arranged together in the correct order that will produce the correct sequence of amino acids of the protein coded for by the transcribed gene. Without this correct stage, the correct protein would not be formed or would not function correctly. To produce the protein, another protein is needed, a ribosome. Ribosome is made of a large subunit and a small subunit which fit together either side of the mRNA strand. The structure of the large ribosome subunit contains space for two tRNA molecules to bind to their complementary anticodon on the mRNA strand. The ribosome then acts like an enzyme as it catalyzes the condensation reaction, forming a peptide bond between the two amino acids on the tRNA molecules to start the formation of a polypeptide chain. All the proteins involved in expression of genes into proteins are essential as the proteins produced control many vital processes within cells such as respiration, which provides energy for living processes such as active transport and movement. Okay, so we've got double bookended importance again. We've got importance at the start, talking about how the expression of genes is really important because it creates specialization in cells, it greatly affects cell structure and function. And then importance at the end, again, talking about why essential proteins control many vital processes in cells, which is a link to the question again as well, because it's talking about the importance of proteins and processes. And then it also links to respiration, which is going to be my next paragraph. This isn't a requirement that every paragraph runs into the next one or links to the next one, but if you can do it, it's lovely, and it makes your essay flow really nicely and really easy to read. Loads of detail in here, and it's A-level detail. We're not doing kind of GCSE level transcription and translation here. We're talking about splicing. We're talking about the activation energy. And also, as you may or may not have noticed, I've got in a lot about enzyme structure here. So we're going to mention enzymes at some point in this essay because they're really important proteins that control loads of processes. So at some point, I had to mention how the structure of enzymes is formed and how that allows them to carry out their function. You only need to do it once, and I've done it at this point. In respiration, many proteins are involved in all stages to produce molecules of ATP, the energy currency of life. During glycolysis, the link reaction and the Krebs cycle, reduced coenzymes NAD and FAD are formed in the cytoplasm and matrix of the mitochondria. These act as electron carriers which transport chemical energy stored as electrons and H+ ions to the electron transport chain. The electron transport chain is a series of cytochrome proteins enfolded in a membrane of the mitochondria known as the cristae. The proteins in the electron transport chain oxidize the reduced NAD and FAD, causing the release of the electrons and the H+ ions. The electrons travel along the ETC in a series of redox reactions, losing energy each time they are transferred between the proteins. The energy lost is transferred into the protein to pumping H+ ions across the membrane from the matrix to the intermembrane space. The number of ATP molecules obtained at the end of the process of oxidative phosphorylation is directly proportional to the number of H+ ions pumped across the membrane, so these proteins play a vital role in the production of ATP. The final electron transport chain protein is the cytochrome oxidase enzyme. This has an active site that binds oxygen, H+ ions, and the electrons to produce a molecule of water. The proton gradient, now formed across the membrane from the intermembrane space to the matrix, is now used to power the enzyme ATP synthase found in the membrane. This structure of this protein acts as a molecular machine. Using the energy from the flow of H+ ions through its channel, it rotates and changes shape, which catalyzes the condensation reaction between ADP and inorganic phosphate to form a molecule of ATP. This process is also used in plants during photophosphorylation, as well as in all eukaryotic and prokaryotic cells, providing a source of energy for all living processes in these cells. Okay, so again, very, very brief sentence at the start to kind of think about the importance. Obviously, it's important because it's producing ATP, and I've mentioned that that's the energy currency for life. That's not really enough. You would need to explain what the energy is used for, which is what I've done then at the bottom. So I've made clear that it's common to a load of different organisms and different processes, and it produces a source of energy for all living processes. You could give examples like movement, active transport, whatever you wanted to do. There could be a nice link to another paragraph there that you could add afterwards, but it's not essential. I've mentioned the proteins I was going to talk about, so the coenzymes, the electron transport chain proteins, and ATP synthase. I've spent specific time talking about the structure and movement, and how the structure of ATP synthase allows it to function as this kind of rotary machine that produces ATP, and that's therefore a nice link into structure and function there. Also, you'll notice that there's a lot of detail here, and it is correct, and it is A-level about respiration, but it's focused on the proteins. I didn't start a glycolysis, go through the link reaction, go through the Krebs cycle, go through oxidative phosphorylation, because that wasn't necessary, and that would have been a waste of my time. Because if you focus on some proteins, this bit where it's the electron transport chain contains the most proteins to talk about. So just focusing on that is more than enough, and it's not worth writing out the whole of respiration and every protein that you come by in order to answer this paragraph. If I was doing a paragraph on photosynthesis, I could have done that as well as this. There's no reason to say you can't do photosynthesis and respiration in the same essay, because they are two very different spec points. But for example, if I did photosynthesis after this, I wouldn't also focus on the electron transport chain. I'd focus on rubisco, probably, because that's a nice, really essential protein to the story of photosynthesis. There's also a nice opportunity there for going beyond the spec and maybe talking about photorespiration to do with rubisco, which is a nice one. Could also talk about pigment proteins. So all the accessory pigments in the photosystem, the chlorophyll as a protein, kind of providing the electrons for the electron transport chain in the first place. There's some other ways that you can do proteins and focus again on a part of photosynthesis that is to do with proteins and isn't exactly the same as this and isn't the whole story. Aerobic respiration in the mitochondria relies on the delivery of oxygen to cells and the removal of carbon dioxide produced. This process is facilitated by the protein hemoglobin. It has quaternary structure due to it being made of 4 polypeptide chains. Each has a prosthetic heme group attached containing an Fe2+ ion. This structure allows the binding of up to 4 oxygen molecules to each hemoglobin protein to form oxyhemoglobin. As each oxygen molecule binds, it changes the shape of the hemoglobin in a way that makes it easier for further oxygen molecules to bind, as it causes a change in tertiary structure, which exposes the Fe2+ binding site. This is known as cooperative binding. This reaction is reversible to allow oxygen to dissociate from the hemoglobin and diffuse into respiring cells. The dissociation of oxygen at respiring tissues is caused by high concentrations of carbon dioxide in the blood plasma. The carbon dioxide enters red blood cells and reacts with water to form carbonic acid. This process is catalyzed by the enzyme carbonic anhydrase. The carbonic acid splits into H+ ions and hydrogen carbonate ions. The low pH caused by the H+ ions alters the tertiary structure of the hemoglobin by disrupting the hydrogen and ionic bonds, which decreases the hemoglobin's affinity for oxygen and makes it easier for it to dissociate. This ensures that oxygen leaves the hemoglobin near cells that require it, as they are clearly carrying out a high rate of respiration. The H+ ions then bind to the hemoglobin to form hemoglobinic acid to prevent the cell from becoming too acidic. Membrane proteins then help to restore the charge of the cell. Carrier proteins actively pump the hydrogen carbon ions out into the blood plasma, and channel proteins allow chloride ions to move into the cell from the blood plasma to compensate. This is known as the chloride shift. Any remaining carbon dioxide which did not go through this process binds to the hemoglobin and is then transported to the lungs. At the lungs, where there is a low partial pressure of carbon dioxide, the hydrogen carbonate ions and H+ ions from the hemoglobin recombine to form carbon dioxide and water, which is then excreted through exhalation. The structure of haemoglobin is vital to its function in the mass transport of gases around the body to be exchanged. Without this process, aerobic respiration would not be able to continue in cells to provide energy for cells, and the pH of blood would not be regulated, which could disrupt other proteins and cell functions. So again, starts and opens with an importance plus a link to the previous paragraph, which was about aerobic respiration. Lots of structural detail about haemoglobin's function here. So I've really got this process and structural importance of the protein kind of down here. All of the green highlighted parts are then my beyond the spec additions, which includes the chloride shift and the carbonic anhydrase. If you took out these green sentences, the rest of what is left would make perfect sense and is perfectly acceptable as the rest of the paragraph. I've added these in so you can see that they're there and how I incorporated it, and see how little I had to incorporate for it to kind of be valid as beyond the spec. However, obviously, it only works if you can remember all this stuff and get it right. And you're only going to know that if you know everything else well enough to be able to add that on top. And then again, I finished with importance. Again, I've got two in here because I've got one that links to the oxygen transport and one that links to the sort of role it plays in functioning in terms of regulating the pH of the blood, but that is linked to my beyond the spec content, so you obviously wouldn't need to put that in. Final paragraph then, which is about immunity. Another group of blood cells that produce important proteins are lymphocytes. These cells are vital in the immune system that defends the body against invading pathogens, as well as destroying abnormal or cancerous cells and tissues. Both T and B lymphocytes have specific membrane proteins which act as receptors for cell recognition and communication. An example is the CD4 receptor on T helper cells, which binds to protein complexes on antigen-presenting cells, cells that have been infected with a virus or a phagocyte that has engulfed a bacterial pathogen, which have placed the pathogen antigens on their cell surface membrane to be activated. Once activated, they will then divide by mitosis and stimulate other T cells and B cells to begin the primary immune response. T helper cells can also produce cell signaling proteins such as interleukins, which help to activate these other cells. Activation of B lymphocytes then stimulates clonal expansion, where they divide by mitosis to form plasma cells and memory B cells. The plasma cells will then produce antibodies. These are proteins that have quaternary structure, that have a variable region with a tertiary structure that is specific to the antigen of the pathogen that triggered the B cell activation. Antibodies work to destroy a pathogen by binding to the antigens on their cell membranes and increasing their chances of being consumed by a phagocyte, or in the case of viruses, preventing their entry into cells so that they can replicate. Memory B and T cells are made for this primary immune response, which means that they contain receptor proteins for the antigens of this pathogen that will be quickly activated again if they bind to the antigens on an invading pathogen in the blood or an infected antigen-presenting cell. This will allow the rapid production of many of these specific antibodies, which will help to destroy the pathogen before it has the chance to replicate and cause symptoms in the body. This allows humans to become immune to certain pathogens and prevents them from suffering if infected again, increasing their chance of survival and reduces the spread of pathogens between individuals, which increases the survival of populations. Okay, so again, got an importance at the start and an importance at the end, and a link to the paragraph before because it says another group of blood cells, so I've linked it from red blood cells to white blood cells, basically. Again, a really tiny thing. It's not something you have to do, but it just feels nice. And then importance at the end again, so talking about the importance of immunity and how that helps increase survival in terms of the secondary immune response, importance of how that helps to prevent the spread of pathogens between individuals. You could have included bits about vaccines here to kind of show that we've sort of harnessed this potential of antibodies and antigens as proteins, and we've kind of used it to help us gain immunity artificially, which would have been a nice kind of way to do this. I've shown you two sentences where I've added a little bit of beyond the spec content, but you can see here they're just like sentences, individual sentences of facts. It's not a whole paragraph, it's not really contributing much, and it's not explaining loads. So this is kind of extra beyond the spec content, not done well and wouldn't necessarily get you that credit. I focused on the structure of antibodies. I said this is why I picked this paragraph. It was a nice one because you can talk about the structure, quaternary structure, specific tertiary structure. As long as you've included that and done it correctly, that's a nice way of making sure you get structure into this paragraph. Yeah, a good one. And again, quite easy, lots of things that you can include, but definitely making it A-level level, talking about antibodies, how they work, the structure of antibodies. the receptor proteins, thinking about things like antigen presenting cells, all of that stuff that you wouldn't have talked about if you were at GCSE, really making sure that you've got that A-level content. Okay, so again, we've got a topic list. It's not exclusive. Again, you can talk about so many things, and there's many things within each of these spec points that you can talk about. So it is definitely not an exclusive list. These are all the things we've mentioned. So again, you can see clearly we definitely talked about more than 4 topics. We had enzymes as catalysts. Within that, we've also, that's a big branching topic. Enzymes controlling metabolic processes, because we did proteins in respiration. We did proteins involved in membrane transport a little bit. We talked about, in some cases, actively pumping things in and out of cells for the hemoglobin. Proteins involved in the immune system. Enzymes involved in DNA replication. Interestingly, hemoglobin isn't necessarily on this list, but metabolic processes, or even just because that counts as excretion. So there's obviously, like we said, ways that you can get proteins in that aren't necessarily going to fit specifically in here, but that would be another spec point as well. It's a nice question, and I like these questions, especially the biochemistry questions, because there's lots of processes you can include, that you can include loads of detailed knowledge and kind of show off, but it has to be correct. So as I said, when you're picking these topics and picking questions, really, really think about how much detail can I go into, and do I know it really well in order to be correct? That's the kind of thing when you're picking these topics, to make sure that you are confident in it. Link to the structure of proteins and how that helps them to count their function. If you went through the whole thing of this and you didn't mention kind of tertiary structure or quaternary structure or shapes fitting together or like the active site, look, that would be a big red flag to an examiner. I don't think it's possible to really get away with not talking about it, but really trying to make sure at least in each topic you mention structure at some point, so you really are answering the question. Link to the importance of what the process is required for and what would happen without it. There's so many things that these proteins are useful for, and if you don't have them, and a knock-on and impact effect. So the importance part for this would have been hopefully fairly easy. Really make sure you put it in there. Put it in at the start, put it in at the end of the paragraph, just to double up and make sure that you know that you've covered your bases. Not too repetitive about enzymes, that's why I tried to pick antibodies and hemoglobin, even though I did mention an enzyme within there. But really try and make sure that even if your whole essay was about enzymes, wouldn't necessarily be wrong, because you'd be covering different spec points, because it'd be the processes that would be different, and then the importance of those processes that would be different. But if you've got something like this where there's so many options, try and be sort of a little bit more general and go for some things that are not just mean you're talking about enzyme all the time. And that would make a kind of nice broad essay answer. But yeah, hopefully you enjoyed it, and yeah, fingers crossed something about proteins comes up in the real exam. Fingers crossed.
Ouch! This is why in some videos I'll explain scratches.